Good morning. Can you hear me okay? Great. My name is John Haddon. I'm Chief Executive and co-founder of Virocell Biologics. I want to thank the Alliance for Regenerative Medicine, and I want to thank you for joining me this morning. So Virocell Biologics is a vector technology company that also has a specialized CDMO that focuses on preclinical and clinical supply exclusively for innovator clients. The core of our experience set stems from the experience that my co-founder, Farzan Farzaneh, who's here, and yesterday co-moderated or moderated the panel on autoimmune disease, derived from about a 15-year experience at King's College London. We characterize what was built there as sort of a mega core vector lab. And over about a 15-year time frame, they manufactured almost 200 GMP batches of viral vector that went into clinical trials around the world. It was about 3/4 lenti and 1/4 gamma retro. And what's little known is that the original way that that team got into business for manufacturing was through vector design. And about 2/3 of that prior experience were novel vector designs. And so the core team at Virocell has had the opportunity to learn from that entire experience. Some worked, some didn't. But in this era of de-risking, we find that clients, both big and small, are very interested in understanding how to de-risk their vector objectively before entering the clinic. So we're headquartered in London. We have about a 70-person team with deep experience across all the categories that you'd expect. So the market need that we're filling here, and we support, from a client perspective, innovators broadly. That includes academics, venture-backed companies, as well as top 10 multinationals, for example. We, generally speaking, meet the client at the preclinical phase, often around this de-risking, vector design de-risking step, which I'll talk about in a moment. And we also provide, in addition to the GMP services, we have both formal and informal relationships with larger CDMOs that have been a fantastic source of client referral over the last couple years. So from a what do we do perspective, I'll talk a little bit more in detail about vector design and optimization. We obviously have process development capabilities as well as GMP. We're focused, as I mentioned, similarly to the way that the team was previously, on lenti and gamma retro. And then I'll touch a little bit on innovation research as well for us. So there are 3 pillars to the innovation research portfolio. First is novel manufacturing technologies. Second are ligand-modified vectors. And third are programmable intelligent vectors. So an example of the output of this team, a subset of which is here in the photograph, is a novel HEC-293 cell line, which performs sort of at or above the market-leading cell line. And we expect that cell line to be both master cell bank and working cell bank by the end of this calendar year, and we'll be pressing that into action in our suspension process. Ligand-modified vectors, as an example, is the concept of adding a ligand to the producer cell such that the virus has that ligand on its surface without modifying the vector genome, so we can help tune that cell to either hone to the right tissue, to have better infection, or potentially for better affinity purification. And then lastly, an example of the programmable intelligent vectors is we're writing code such that we can develop, for example, a safety switch that's auto-regulated, such that if the cell that's been modified by the vector begins the process of malignant transformation, an apoptotic signal can be initiated, such that that cell is then prevented from completing the process. I think we need a new battery. So how do we help a client de-risk? So it's a collaborative process, obviously. We will work through with the client, a concept structure such that we look at various promoters, various enhancing sequences, various regulatory sequences, and other. But instead of making an opinion-based recommendation, we prefer to make a data-driven decision with the client And so what we do is the microbatch offering is anywhere from 2 to 20 batches. These are small, 150 to 300 mils. But we look at the effect in the target cell. So collectively, we can make a decision to say, if this is the product profile that you want to deliver, this or these two, for example, of the mix are the best in doing that. And that way, the client can, with confidence, look at entering the clinic. We have a passionate team, and Kesha is photographed here, focused on eking out any element of increased yield that's possible. I think what's very clear is in this environment, there are a couple themes. There's significant need for de-risking, but everybody wants higher titer at the same time that they're asking the vector to do more or also deliver a larger payload. And as you all know, that has a downward impact on titer. And so our team is focused on maximizing titers for your clinical application. We manufacture our GMP product in central London. The facility is located at the Zayed Center for Research. This is a facility that is both MHRA inspected. Its GMP certificate was issued last year. It is also licensed for vector manufacture and export. So we're able to export globally to support your clinical trial needs wherever those might be. So to wrap up, we're ready to join your journey wherever you need help, whether it's as early as having identified the gene of interest and in need of making sure that it does what it's supposed to do, or later in the process. But we have no ambition of being a commercial scale manufacturer. We want to help take you through that journey through the clinical trial process. And with that, members of the team are here today. My hope is that we have a chance to kind of get back on time. And thank you for joining us this third day, and happy to take any questions if you have any. Cheers. Okay, you got 7 minutes back on your day.